Carlos, for starting a little late. Carlos Santibáñez López is joining us from across the pond. He's an assistant professor at the Western Connecticut State University. And Carlos started his scientific career in Mexico. He did his undergraduate work and his PhD thesis and his first postdoc, I believe, at the National Autonomous University of Mexico in Mexico City. Afterwards, he did two postdoctoral positions, one at University of Wisconsin-Madison and another at Eastern Connecticut State University, before landing at Western Connecticut State University, from where he's joining us today. So Carlos works on, broadly speaking, the evolutionary biology of arachnids. But I think looking at his publications, there's one group that makes his heart flutter more than others, which is scorpions. And this is what he will be talking to us about today. So thank you, Carlos, for joining us today. The digital podium is yours.
Thank you so much, Ronald. I'm quite excited, and this is such an honor for me to be here with you. Let me just begin by sharing my screen, and please confirm that you are indeed looking at my presentation.
Yep,
Slides.
I can see your slide.
Perfect. Well, without any further, let me just tell you that today I'm going to talk a little bit about what my lab has been doing in the past 2 years. Ivan said something interesting, and this is about COVID, right? COVID broke a lot of these research, and therefore it has just been delayed a lot of the results, including the annotation of the first urate scorpion that I'm going to briefly mention at the end of my talk. So, sorry, I do believe that it's interesting just to give a quick introduction about the organisms that I'm working with. And these are the scorpions. You may be very familiar with them. They are quite charismatic, and they have been pushed as a threat and in the media. They are an old lineage. They have a wide distribution, but they are not like the spiders that are like very diverse in forms, in shapes. These animals have a very conserved morphology. You can find them literally everywhere except in some of the boreal areas, the poles, and some of oceanic islands, unfortunately. Depending on the authority, you can recognize between 22, 23 families, about 225 genera, and less than 3,000 species. Throughout this talk, I'm going to refer to two major groups of scorpions. The first one is known as Butida. It's called parvorder. It's just a taxonomical category that normally we use for some of these organisms. What I want you to remember from these two groups, it's not probably the name, but I'm just going to use some colors to illustrate when I'm talking about these major groups. So orange will be always referring to the Butida group, which includes already 3 families. Among them, family Butidae, that some of you may be familiar with. This family contains most of the highly toxic scorpions to mammals. And currently we can recognize 102 genera and half of the diversity of these species. The other major group that I want you to talk about today is the parvorder Urida, that contains the other half of the diversity. And as you can see here in this slide, it definitely includes more families than the other group. We currently recognize 20 families and about a little bit more genera than the other one. And this is where you're going to find all the other scorpions. We recognized some species that are highly toxic to mammals, and these are included in the genus Hemiscorpius, which is distributed in the Middle East. But the venom is slightly different than Butids. It is important for me just to to make a quick comment on the phylogenetic position of scorpions within the chelicerates or arachnids, because it tells us a little bit how the venom has evolved in these organisms. So on your screen, you're looking at different pictures of the relatives to scorpions. That goes from spiders and ticks all the way to now the horseshoe crabs, camel spiders, sea scorpions, and so on. And it is important because in the last few years, we have tried to uncover the evolutionary history of these chelicerates. Our latest topology suggests that horseshoe crabs are included in these arachnids, and therefore it changes the scenario of a possible unique colonization of land of these animals. It is important also because through the use of other type of data, like developmental data, we have supported the idea that scorpions are a derived group and not the sister group to the rest of the arachnid, like it used to be acknowledged back in the 70s. So on your screen, we have some pictures of scorpion embryos with in situ hybridization of some genes. This work was conducted by one of my students, one of my mentees while I was at University of Wisconsin, Eric Nolan, and we published this, and it was quite interesting because we show that spiders and scorpions share these duplication of these genes and similar functions. And then later on, Andrew Ontano, a PhD student at Sharma Lab, used pseudoscorpion genome and search for some of these genomic rare events that we're calling to find duplications of these Hox genes to further support the idea that pseudoscorpions are also a derived group. And more importantly, in this work, we propose the idea that scorpions and pseudoscorpions are sister group to each other. And this is mainly because there are some characters that can support this, including the courtship and the presence of venom, which is technically the reason why I'm talking about this, because it seems like the venom origins in this clade that we call Arachnopulmonata, that includes spiders, pseudoscorpions, and scorpions, 3 of the 4 venomous groups in chelicerates, may have had a unique origin. And this is one hypothesis that we explored very briefly in the first venom transcriptome of a pseudoscorpion species. And then we just came up with some search of orthologus and paralogus of some of the most common venom components that we thought that they may be sharing between pseudoscorpions, scorpions, and spiders. And so we tried to pinpoint the origin of some of these genes in each of these lineages. But of course, spiders are not my area of expertise, and I cannot comment on those. It's quite impressive the amount of work that has have been done in spider venom evolution, and I encourage you to go to the proper literature. But today I want to focus on scorpions. And so venom plays key roles in the scorpion biology of, and we can see that it is used definitely to hunt their prey. Unfortunately, it is also used as a mechanism of defense. And in this case, given our membership to mammals, and mammals being most of the predators in these animals, that venom affects us greatly. And the other role, or the main role of the venom in scorpion biology, is courtship. So we have seen some species stinging, some males stinging females during the courtship, and it may have some specific roles that we have not uncovered yet, but we're currently taking a look at it. So today I'm just going to talk about 2 questions that we have been trying to answer since my first postdoc. One of them is that I'm interested in understanding the origin of these scorpion toxins, and I'm going to talk about specifically some of them. And the other one is if there's a different scorpion lineages exhibit venom phenotypes, specifically because now, given the current phylogenetic hypothesis of these animals, contradicts all the major morphological characteristics that diagnosed scorpion groups in the past. So to address the first one, let me take you a little bit through what we have been doing. The first thing, and I think I thank Ivan for mentioning this too, it is important for us to have a solid phylogenetic scenario, an evolutionary origin of these animals. And so on your screen, we have the latest biology of scorpions using ultra-conserved elements. And in this publication, we test the differences between the topologies recovered using UC yeast and amino acids. And technically, the topology seems to be solid. Again, all I want you to remember is the colors. Orange is one group, as you can see, is the sister group to the purple. Again, the orange clade includes only 3 families. The purple clade includes the rest of this thing. So as you can see, this is solid, and we're happy that nothing has changed. And then what the other thing that we need to do is search for these different components using venom transcriptomes. And this is something that I did in my first postdoc. And I'm sorry to say, Ronald, I did 3. The first one was actually at the Posani lab at the Biotechnology Institute of the National Autonomous University of Mexico. And you know, Posani is one of the big names in scorpion research. And so while I was there, I set myself one thing. At that time, most of these venom transcriptomes were conducted using butid species, right? Yes, it's definitely understandable. These things are the most toxic to humans, and we need to find out the differences between the composition of these animals. However, as I showed you already, in that big phylogeny, we have 23 families that were neglected at that time. And so one of my tasks was to take the advantage of Mexico, and because it's one of the most diverse countries in terms of scorpion diversity. And I decided to go out and collect different animals from different families just to take a look at the venom compositions. And so on your screen, I'm just looking at some charts. You're looking at some charts of the different compositions of these different organisms. And when I came back to the US to do my first postdoc, I had the opportunity to get access to more and more transcriptomes. Some of these transcriptomes were not sequenced from the venom glands, but some other scorpion parts. And so I had the opportunity to build a more strong phylogeny at that time. But at the same time, I was looking at some interesting patterns that came across during my first postdoc, and that was the presence or the expression of one particular type of peptides known as calcins. These calcins are ryanodine receptors. They have this affinity for these receptors, and in the literature, they were only reported from urid scorpions. Butids didn't show that, and throughout the survey of transcriptomes, I noticed that it was indeed they were not expressed. Now we know that transcriptomes are just showing you expression of specific genes, but the lack of expression doesn't deny the presence in the genome. So all we said here is that these scorpions are not expressing calcins. However, we noticed that these butid scorpions were indeed expressing something else, and that was lambda potassium toxins. So these toxins were affecting potassium channels. They shared the ICK, the inhibitor cysteine NOT motif, and therefore we thought that they would maybe somehow related to each other. They may be paralogous, orthologous, or something of that sort. So we came up with the mining the transcriptomes at that time. We retrieved all that information. We ran phylogenetic analysis and some sort of peptide clustering. And we found indeed that the LKTXs were expressed only in butids, and the calcins were also expressed on urids. However, one day while I was looking at my window, it was snowing, it was amazing. I remember my high school days, and I remember that it was somehow a little bit of annoying that we tend to express the chemical formula with letters, And so I said to myself, well, these things must have a 3D structure, and they do indeed, right? Whenever they are in a solution. So why don't we use that structure to analyze it, just to use the shape of this thing and see if there are some differences between these two major toxins. And so what you're looking at that is that structure. It was reduced to obviously 2 sites only. At the time, I didn't know if we had the possibility to explore it in a 3-dimensional way. So on your left, you have the frontal, something that I call the frontal shape, and it was given by the positions of a specific amino acids. And on your right, you have the lateral view. And so this is a principal component analysis of these different shapes, and it's the phylogeny of the scorpions plotted onto this morphospace. And so we clearly see these differences. And so this 3D structure of these two components that were restricted to these two major groups of scorpions are following this position history. And so there we see that calcines are restricted to urida. And when we incorporate more information about the chemical and physiological properties of these molecules, we see that indeed they are unique to urida. More or less, we explored the differences between the shapes. We studied the evolutionary pressures, and we see that those sites that are indeed the ones that are recognized in the receptors, specifically in calcines, they are evolving under negative selection. But of course, that's not also, we didn't stop there. We try to understand if these calcines were also following scorpion diversification within the urida, within this major purple clade, and indeed it was. At that time, these two families, color in magenta and purple, were considered as sister groups. Here, our phylogeny didn't recover them together, and more importantly, the shape of these different toxins were separate them in the principal component analysis. All right, so years in the future, we had more access to more transcriptomes, and this is a work that we published just a year ago, where we explored 100 scorpion transcriptomes. And so we mine for the most important components. In this case, things that we recognize as toxins. So you see on this chart, on this slide, a heat map of those components. And we're just counting number of transcripts or proteins from these different species. Here we have the Butida. This is the orange clade. And as you can see, the sodium toxins are by far more abundant in here. And so if we compare that against the purple clade, yes, we have here some colors, but again, just take a look at the scale. So we're talking about 0 to 7 transcripts of proteins. In the previous slides, we're just looking at 0 to 38. So indeed, there are more sodium toxins present in the other group. And again, we observe that calcines are unique here. They are not present in the orange clade. And also, chloride toxins seems to be restricted to some specific butid lineage. And I'm going to talk about them later. So uncovering the origin of these toxins, specifically the cysteine stabilized alpha beta motif scorpion toxins, is very challenging because we're talking about a sequence of peptides that are like the longest is about 120 amino acids. So running phylogenetic gene trees, it's just very challenging. So what you're looking on your screen is, again, the analysis of thousands of sequences of these scorpion toxins retrieved from the literature and including our transcriptomes. In pink, we see those that had certain affinity for the sodium toxins channels, including some transcriptomes, and some that have their function review. Moreover, we see that the potassium, they also are independent clade from this, along with the chloride, the calcines, and the DDH, which is the disulfide other calcine toxins. I'm sorry, I'm rushing through these results because they're technically important, but I'm going to address them later in a different slide. So if I zoom into the potassium, we also observe that some of the current classification of these toxins is recovered, except for some specific case like the beta potassium toxins. We have two clades, one that was unique to butyds that were recovered, nested within something that we call the S-corpine, which are also known as orphan peptides. And again, we observe some specific toxins restricted to specific groups, like the lambda potassium toxins that we addressed already, and the kappa potassium toxins that are restricted to urics, which I'm going to show you later. Of course, we were also interested in understanding the origin of the sodium toxins, because those are the ones that affect greatly to mammals, specifically humans. And again, just coming up with a gene tree for all of these peptides seems to be very challenging. And of course, sometimes, depending on the model that we're using, you could recover different hypotheses. So what you're looking at here is, again, the analysis of 800 sequences of these peptides. The colors are indicating the affinity. In green, we have those sodium toxins that only affect arthropods. In red, you only see those affecting mammals. And in blue, you see those affecting both. And so we are just confirming that the origin of these sodium toxins that affect mammals have independent origins, and they are not restricted just to a single one. And to double check this hypothesis, we used a different methodology. This is just the clans, this is clustering using similarity between the peptides. And as you can see, again, we observe different clusters of these peptides depending on their function. So again, we support the idea of these mammalian sodium toxins having independent origins. And using a different method, because just try to date the origin of these with molecular techniques was a little bit challenging. We came up with the idea of dating the scorpions that contains these different toxins. And so you see here that the origin of these different groups that possess this specific sodium toxins more or less is contemporaneous to their predators. So on your screen, you have a classic image of the grasshopper mouse, that eats the Arizona bark scorpion, which of course is one of the deadly scorpions here in the US. So with this approach, we more or less try to pinpoint the origin of these toxins in the scorpion tree of life. All right, so this is something that we still studying, and for that we're going to need genomes, and I'm going to talk about that in the last slides. But before going into there, let me just briefly mention about these phenotypes. Now, scorpions, they exhibit some morphological adaptations that are specific to the type of the microhabitats that they live, right? So we have scorpions that lives in the crevices, and therefore they look like a tortilla, right? So they are dorsally, ventrally compressed. Then you have other scorpions, like those that dig burrows with powerful pincers and powerful chelicera to remove the soil. Scorpions that inhabit the deepest caves in the world, like this one here on the bottom, that lacks eyes and pigmentations, and they have elongated pedipods. So one of my questions is that is venom somehow related to these ecomorphotypes, to these morphological adaptations? And so we try to come up with this idea back in 2018. Jimena, she was a PhD student at Posani's lab, and so she did this comparative diversity between the transcripts of these different scorpion species and tried to correlate it with the ecomorphotype. We couldn't observe too much. As you can see here, Centauroides still just has the highest number of sodium toxins, because yes, that's a species that is highly toxic, but it doesn't tell you too much about the different ecomorphotypes. Instead, we're now trying to study more microhabitats and try to come up with more data to support that it may be there might be some differences in the venom compositions. So two of my students are currently studying the venom composition of two interesting species. One on your left is a Brachystosternus from Argentina, and the species inhabits these mountains that goes from sea level altitude all the way to 4,000 meters above the sea level. And the other one, Europhonius, is a very interesting species because it has winter activity. That means that this animal is only active during snow, and so it will be very interesting to understand the venom composition of these animals. Also, another student, Adam is building this machine learning, this artificial intelligence, scripts to study the venom composition, try to come up with the idea that we can further down the classification of these venom components into phenotypes. And so what we see here is the principal component analysis of only, this is not a expression levels, this is just a number of transcripts again. And there's a reason why. Most of these transcriptomes were generated throughout differences in techniques and different labs. So there is no standardization to count just the transcripts. So on your screen, we're only looking at number of transcripts. And again, we're looking at the different colors, purple and orange. And I think, and so I'm just using 4 classifications. Those toxins that have affinity for the potassium channel, those affecting sodium, those affecting chloride, and those affecting calciums. And as you can see, the putative venom phenotype for urids, it's more, it is diagnosed for the inclusion of more toxins that affect calciums than the venom phenotype for the buthids. And so if we further explore more into a subclassification of these different categories, we observed again like a breakdown in this between these two phenotypes, with urids expressing calciums, VDH, one specific type of sodium toxins that is only affecting insects, and ascorpines, which are these toxins that are known as orphan, because they also affect potassium channels, but again, they may have antimicrobial activity as well. And then we observe the other ones, which are loaded with sodium toxins, some other potassium types, and yes, just specifically those two. This is preliminary results from this study that Adam is conducting right now, and I hope that in the following months we could have more results, more interesting results. And so just to sum up and to wrap up this talk, we definitely need scorpion genomes, and this is just a table that shows you the disparity between the availability of genomes of other organisms or chelicerate organisms like spiders. We have up to 43 genomes. Some of them are already at the level of chromosomes. We have only one genome for harvestment, one genome for a pseudoscorpion, which actually now it's at the level of chromosome. And if we just take a look at the scorpions, we have 4 genomes of 4 species within the butid family. None of those are at the level of chromosomes, and most of them are highly fragmented. I think the latest one, which is Centauroides vittatus, is perhaps the most stable. It was just recently published. And then what we're doing right now is sequencing 2 species of Carillus. And again, these are preliminary data. We are assembling, but we project that these species may have a 3.1 to 4.2 gigabases. So this is a very impressive genome compared to the ones that we have for the family Buthidae. And the one that we have currently at the end of the annotation, in fact, I just got the message from my collaborators at UConn that's saying that the annotation is almost done. It's the Herdurus arizonensis, which is a species of the Urida clade, and currently the size is 2.2 gigabases with 915 contigs. These will allow us to do some comparative analysis like those presented by Ivan, which I think it's by far the better one, just to pinpoint the origin of these things and have a better picture of scorpion venom evolution. And we're also going to use the other genomes, spiders, still scorpions, and we're currently sequence also the rest of the arachnids, just to have a better idea on how to search for these putative duplications of these different genes that may result in the origin of these different toxins. And so without, I just want to acknowledge a bunch of people, my collaborators at the Posani lab, the Sharma lab at University of Wisconsin, Graham lab here at Eastern, and of course, all my students that without their support, none of this could be possible. And so thank you so much for your attention. And if there's time for some questions, I am happy to take them.
Thank you very much, Carlos. Very interesting. The situation with the